Hello class, this is Dr. Anderson again, and we are continuing our journey in Clinical Physiology 1, and this presentation is on the topic of homeostasis. So we start with learning objectives. I want you to be able to define homeostasis in your own words, and you need to be thinking about what the internal environment is and how the internal environment is being regulated and what kind of things are regulated by homeostasis. So the internal environment involves the body compartments. So we have intracellular fluid and extracellular fluid. And extracellular fluid is divided into plasma and interstitial fluid. So what's the composition of these compartments? Which one's the biggest? Which one's the smallest? Which one is directly regulated by homeostasis? So what is a feedback loop? How does feedback control homeostatic parameters? And sometimes we talk about positive feedback, but mostly we're talking about negative feedback. Homeostasis is a variable kind of thing. It's influenced by what's going on with that particular individual person. And so I think of homeostasis on a continuum. The more people are maintaining homeostasis, the healthier they are. The less able they are to maintain homeostasis, the sicker they are. I think homeostasis works very well in that terms of a framework. However, it's also important to realize that homeostasis varies depending on biological rhythms. So we have a master pacemaker in the suprachiasmatic nucleus of the hypothalamus. That hypothalamus is aware of day and dark and light cycles. We have sensors that are aware of our what we eat, our activity, those kinds of things, and those are influencing the rhythms that we exhibit. And it's important to think about rhythms because we think about homeostasis as a process that can respond to a biological stress. When you overlay the idea of biological rhythms actually anticipate biological responses. Biological rhythms anticipate that we're going to get up and be active during the day, or that we're going to lie down and sleep during the night, and that kind of thing. So it's important that you be able to synthesize those two concepts together. So homeostasis is a dynamic process by which the environment of the cell is maintained so that the cell is able to survive. So we in physiology think about something called cell theory. We're made of cells, and the cells have to be healthy, and the cells have to be working together in a healthy way for the organism to be healthy. Sometimes people describe homeostasis as an equilibrium. Sometimes you'll even see that gasp in textbooks, and that just isn't true. Homeostasis is a steady state. When something is left alone and left to reach equilibrium, that's not life. Human beings that are in equilibrium are dead. So I want you to think about homeostasis as a steady state. It's very dynamic, give and take. The internal environment is the environment that the cells live in, and the fluid that the cells live in are the extracellular fluid. So as homeostasis is conceived as a framework, we identify the extracellular fluid as the internal environment. If we regulate the outside of the cell, then we assume that the inside of the cell will be regulated indirectly. So we may do this in class. Take a moment to think to yourself. You can even pause the presentation. Think about what cells need in order to survive. We'll also be doing an exercise in class to get you to think about this. So here are our fluid compartments. Notice that we have an intracellular compartment that's 28 liters in a typical person, or 2/3 of the total body water. So most of your fluid is inside the cells. Even though one cell is really, really tiny, when you add up all the billions of cells, you have a very large fluid component. Directly surrounding the cells is the interstitium, and then the interstitium is in communication with the plasma which is a very small part of the extracellular fluid, though that's where a lot of the regulation goes. So I want you to think about, if you ingest water, that water is first going to go into the capillary. It's going to distribute between the capillary and the interstitium, and then will distribute between the interstitium and the inside of the cell. So the water you ingest eventually could make it to the inside of a cell, but it has to go through the plasma, then the interstitium, and that takes some time. If you ingest some sodium, that sodium could go into the plasma, it could go in the interstitium, but your cells turn around and pump out the sodium, so sodium wouldn't necessarily have access to the inside of a cell. Those are some of the things we think about when we think about fluid compartments, and we'll come back to these ideas as we go to the cardiovascular system and the renal system and those things during this course. So homeostasis relies on feedback mechanisms, so we need to think about the basics of a feedback loop. If you have something going on in the environment, you have to be able to monitor that, and you monitor that with a receptor. So a change in some kind of parameter is the stimulus. The receptor is aware of that. The receptor then uses a pathway to communicate with an integrating center or a control center. The control center then activates an outgoing pathway or an efferent pathway. The effector then carries out the response. The effector could be a gland. The effector could be a muscle. The effector could be a single cell. So we can think about neural mechanisms. You have a stimulus, you have a neural receptor, you have a sensory neuron, you have something in the spinal cord of the brain, a motor pathway, a muscle, some kind of response. Or we could have all of this in a single cell. You know, the glucose goes up, the beta cell detects an increase in glucose, realizes a response is needed, releases insulin then goes out and does something about the glucose system. So feedback loops are, we have many varieties of those in the body. So when we talk about feedback loops, we can talk about intrinsic versus extrinsic mechanisms. An intrinsic mechanism is one that if you removed the cell or the tissue from the body, provided it with physiologic conditions, you would still see the mechanism. So an example of this is autoregulation. Pressure changes in a brain blood vessel result in dilation or constriction wherever that's appropriate, and that would happen inside the body or outside the body. Autoregulation also happens in the kidney. You take a rabbit kidney out of the rabbit and you perfuse it, you will still see autoregulation. So it's called an intrinsic mechanism because it is intrinsic to the tissue. Intrinsic means you need outside sources for regulation, meaning either there's a neuron or a hormone involved in the pathway. And so you have to be, the tissue has to be in the organism. So an intrinsic mechanism, you take the kidney out, and now the sympathetics can't affect the kidney, and now you don't have an extrinsic regulation of blood flow in the kidney, for example. So we have combinations of intrinsic and extrinsic mechanisms that we will talk about in this course, and we have combinations of negative feedback and positive feedback. The feedback, the way you figure out the feedback is the relationship between the stimulus and the response. If you have an increasing stimulus and the reaction of the body is to decrease, in other words, the response opposes the stimulus, that's negative. If you have an increase in the stimulus and the response is to increase it even more, that's a positive feedback mechanism. So temperature regulation is an excellent example of a negative feedback mechanism, and in this particular picture we see temperature homeostasis in a person, and we see regulating temperature in somebody's house. So here's a person, they're out hiking, it starts to snow, their receptors detect the low temperature. Sensory information is sent to the hypothalamus, which is where the integrator is. Then the integrator activates motor neurons to the muscles, and the person starts to shiver to generate heat. So the stimulus is cold, the response is to generate heat. And so this is a flow chart demonstration of this. The temperature goes down, the person loses heat, the body temperature goes down. And the response is to do things to create heat. Shiver, curl up, vasoconstriction, trying to return the temperature back to set point. Positive feedback, so we have contractions because the baby's head is pushing on the cervix. That stretches the cervix. That causes release of oxytocin and more contractions, which causes more stretch, and more stretch of the cervix, and more activation of the brain, the hypothalamus, and more release of oxytocin, and more contractions. And this keeps building and building and building until you reach some kind of completion, which is the birth of the baby and the birth of the placenta. Now, homeostasis can vary from individual to individual. There are lots of things that influence your ability to maintain homeostasis. Your age. It is harder to maintain homeostasis as you get older. There are differences between men and women in terms of homeostasis. Your genetics will influence how you maintain homeostasis. Some people don't maintain their blood pressure as others would because of genetics. Depends on how big the stress is. I mean, if you have a really awful bacterial infection, your body may not be able to maintain homeostasis. If you lose a little bit of blood, you may maintain blood pressure. If you lose a whole lot of blood, you may not be able to. It can be the context, time of the day, the culture that you're in, the place that you're in. You know, a person may be able to withstand a stress if they're in their own home, but if they're they're on vacation and they're away from home and the extra stress, they may not be able to. We also have to think about the influence of biological rhythms. So biological rhythms are internal mechanisms, and the beauty of them is that they anticipate stimuli. Our major clock, or our major integrator, is in the suprachiasmatic nucleus of the hypothalamus. So as we are exposed to daylight and darkness, you know, this is why you're not supposed to be looking at computer screens right before you go to bed, because you're stimulating the suprachiasmatic nucleus in a way that is going to interfere with your ability to sleep. These rhythms are based on clock genes, and these clock genes have a circadian expression. So circadian means about a day, and so circadian rhythm, actually the circadian rhythm was a term that was invented by a scientist by the name of Dr. Franz Hallberg. Dr. Franz Hallberg founded the Hallberg Chronobiology Center, which is right here at the University of Minnesota. So a lot of work with biological rhythms taking place here. So for example, BMAL1 and PER2 are genes that are expressed, their expression changes on a 24-hour cycle. So we just talked about temperature as being regulated by homeostasis and negative feedback mechanism, but it also has a circadian rhythm. So here's the idea about response and anticipation. If your temperature goes down, you do things to heat yourself up to get back to your set point. Or if your temperature goes up, you do things to cool yourself. Those are corrective actions. But your temperature is varying over a 24-hour cycle with an increase early in the morning. And let me show you what that looks like. So here we see temperature goes up in the morning and it fluctuates a little bit. Usually we have the highest temperature around in the afternoon somewhere. And then this person goes to bed, you turn the lights off, the temperature goes back down, it stays down. But look at what's happening here and a little bit that's happening here. Do you see the temperature is going up before the person gets up? So the person's alarm goes on at 6 a.m. and they get up, but notice how the temperature is increasing before they get off. there's a biological rhythm that anticipates getting up for the day, and we have increases in temperature. We see a similar thing with a hormone called cortisol. Cortisol is secreted in response to stress, but cortisol is also secreted with a circadian rhythm, and we see cortisol going up early in the morning, as if that rhythm is preparing us for the stress of the day. It's just really beautiful how these things fit together. So all tissues have rhythms with normal variability. There's a rhythm in our hypertension, and there's a variability in that hypertension. And let me just back up a little bit. All kinds of things have variability. Your heart rate has variability. Your temperature has variability. Whoops, I'm going the wrong way. See how the temperature just doesn't stay there. It's fluctuating all over the place. So variability is a good thing. Variability says something about your body's ability to anticipate and to respond to stressors. So babies in the NICU, you want to see variability in their heart rate. If you have a baby and their heart rate is rock solid 100 beats a minute and it doesn't change, you're worried about that baby, because the baby doesn't have any variability. It doesn't have an ability to respond. That's a worrisome sign. See that at the end of life people with heart failure. When their heart rate becomes rock solid and is not varying, it means they've lost their ability for homeostasis and they've lost their biological rhythms. And that has very severe consequences. So hypertension varies, and if you have an abnormal variability, it increases your risk. So if you have hypertension with normal variability, you're at less risk than somebody with hypertension and abnormal variability. And so there's a professor at the U who's very interested in these rhythms, and he believes that patients may need to tailor when they take their antihypertensives to their biological rhythms to get better management of their hypertension. This is really, I really need you to be aware of how important biological rhythms are in the practice of medicine and the practice of other healthcare fields, nursing as well. So abnormal rhythms are implicated in diabetes, obesity, hypertension, cardiovascular disease, kidney disease, and lots of these researchers are thinking about what's going on with the rhythm. There's another researcher at the U of M who's studying obesity, and her program of research is she's using restrictive feeding. She says to her patients, you can eat what you want during these hours, but during these hours you need to be fasting. So periods of eating and a good period of fasting is evidently necessary to good metabolism and good weight management. And so that's the focus of her research, and she's finding some success. So I just want you to be aware that this isn't a futuristic thing. This is happening in research right now. So think about your questions. Go back and look at those learning objectives. If there's a learning objective and you don't know what it means, you need to be asking me. If there's a learning objective and you're not sure how to answer it, you need to be talking in class or asking questions on the forum and that sort of thing. So I thank you for your attention. I hope things are going well so far, and I will see you next time.